All right, we are going to talk about variables in experiments and how we graph with those variables and how we can use that to write hypotheses. So first we have to know, what is a variable? So a variable is a factor that you can change in an experiment. And there's many different types of variables, and we're going to go through those four important types for us. And we're going to talk about how only changing certain things in an experiment is going to allow you to collect better data. So the first variable we're going to talk about is your independent variable. So your independent variable in the experiment is the variable that the scientist, you the scientist, is going to change on purpose to see what effect it's going to have. So your independent variable is sometimes known as your manipulated variable. These are the things we're changing on purpose. We only want to change one thing. We only want to have one independent variable so that we know exactly what that thing causes the result of, what happens. So for example, if we're trying to figure out how quickly water boils, we could alter the amount of water in a pot or the temperature, but we don't want to do both. We just want to do one thing. Your dependent variable is what is going to happen as a result of the independent variable. Your dependent variable depends on the independent variable. So this is also known as your responding variable. It's going to respond to the manipulation from the independent variable. Again, we only want to focus on one thing. So for example, we can examine how much sugar can dissolve in a certain volume of water at various temperatures. So we're going to alter the temperature, which is your independent variable, and see how it affects how much sugar can be dissolved, which would be your dependent variable. Your next variable would be your controlled variable. So control variable, also sometimes known as a control group, is almost like a second independent variable, but not, it is there to see what would happen if we did no manipulations at all. So we can see exactly what the relationship between the independent variable and the dependent variable is. For example, if we want to examine the relationship between how much sunlight plants receive and how tall they grow, we'd want to make sure that nothing else changes. We want to see what would happen if we had a plant that received no sunlight, because a plant's going to grow whether it has sunlight or not, we think. So we want to know what happens if there's no sunlight at all. So that's why we'd want a control group. The next variable is a constant variable. So a constant variable is any other aspect of the experiment that you're going to want to keep the same on purpose all the time. So for example, if Jimmy's having his vision tested by reading an eye chart and he stands 6 feet back from the chart for all of his tests while the doctor changes the sizes of the letter. If Jimmy stands in the same position for all the tests, it's a constant variable, because regardless of how big or small the letters are, he is still standing in the same spot, and that's going to allow him to say, I can read these letters when you change the sizes of them because I'm staying in the same position. So the next thing we'll talk about is graphing data. When we graph data, there's important components to have your graph. We want to have a title in which we state the purpose of the graph, including the variables. We want to use dry mix, which we'll talk about more on the next slide. We want to make sure we label our axis with appropriate labels that tell you exactly what we're measuring. And we want to use scales that make sense. If I'm measuring temperatures from 0 degrees Celsius to 25 degrees Celsius, I'm probably want to go in increments of 5. So 0, 5, 10, 15, 20, 25. I'm not going to want to have my scale from 0 to 60 degrees Celsius going in tens. That's going to be way too big and not show enough detail. And then also on my graph, if I'm showing more than one thing, I want to make sure I include some sort of key that explains what's being shown. When I look at a graph, I'm going to want to be able to say, this is what was manipulated in this experiment, and this is the result from that experiment, just by looking at the graph. So when we talk about dry mix, this is telling us how to graph data on our graph. So dry mix means dependent responding variable goes on your y-axis. Manipulated independent variable goes on your x-axis. So dry mix helps us to remember where we graph our data. The reason why we do all this stuff in science is so that we can replicate our data and be able to use it again. We want someone else to be able to repeat our experiment and get similar results, And if we have all of our variables set up the same way and everything makes sense logically, then someone else can check to make sure that what we did wasn't a fluke. It makes sense. So that's the reason why we do these things. So let's do some examples of identifying our variables. I'm flying across the screen. Come on, keep going. There we go. All right, and I'm going to color coordinate because I obviously am not good at writing on the screen for this stuff. So what we're going to do is identify your independent variable, your dependent variable, and your controlled variable. If this will let me select my color, that would be great. Okay, so independent variable. It says you water 3 sunflower plants with water every 2 days. Each plant receives a different amount of water. One plant gets 6 cups, one plant gets 12 cups, one plant gets 24 cups of water. After a 2-week period, the height is measured. So what are we manipulating? We are manipulating the amount of water because each plant is receiving a different amount of water. Okay, what are we measuring as a result of the amount of water? What we're measuring as a result of that is height. So how tall the plant grows. And what are we keeping the same on purpose during all of this? Well, it looks like we're keeping the same, that we have sunflower plants. They're going to get water. Oh, we're watering them every 2 days. And it looks like it's taking place over a 2-week period. So these are all things we're keeping the same on purpose. And I have that written out on the next slide. So sunflower plants with water every 2 days for a 2-week period is your controlled. Your dependent variable is the height that's measured after 2 weeks. And then your independent variable is the amount of water each plant receives. So let's do another one. In this one, we're going to also identify a control group. So you're testing how much sunlight affects the amount of flowers in a particular kind of rose bush. You plant 4 rose bushes. Each rose bush is put in direct sunlight for a different amount of hours every day. Rose bush 1 gets 12 hours, rose bush 2 gets 6, rose bush 3 gets 3, and rose bush 4 gets no sunlight. You keep all the plants in a climate-controlled greenhouse where each plant is kept at 72 degrees Fahrenheit and watered twice daily with 2 cups of water. After 1 month, you count the number of flowers on each rose bush. So this is a lot to unpack, but first, what are we manipulating? We are manipulating how much sunlight affects the amount of flowers. So how much sunlight? Okay. Come on. All right. Now, what is going to happen as a result of the amount of sunlight? What are we measuring as a result of that? Oh, I changed my colors for some reason. This should be red now. So what's happening as a result of the amount of sunlight? We are looking at the number of flowers that occur on each rose bush. So the number of flowers, the amount of flowers that grow. All right, what are we keeping the same through our constants? Let's see, we have a particular type. So we're going to say they're all the same kind of rose bush. They're all put in direct sunlight, but that was how much sunlight? And that's every day we're doing this, okay? It's in a climate-controlled greenhouse. They're kept at 70 degrees Fahrenheit, watered twice daily with 2 cups of water. Now, we're now looking for our control group. Control group is going to be the one in the experiment that doesn't get anything. It's the one that we're measuring everything against to see what happens if we don't change the independent variable. So that would be rose bush number 4 that gets no sunlight. Oop, let's get back. There we go. So, amount of sunlight is going to be your independent variable. Your number of flowers that grow is your dependent. Your constants are the climate-controlled greenhouse, 72 degrees Fahrenheit, watered twice daily with 2 cups of water, all the same kind of rose bush. And your control group is that rose bush 4 gets no sunlight. So now we're going to write a hypothesis based on this experiment. So a scientific hypothesis has to meet 2 criteria. It has to be testable. It's got to be something that we can test and observe those tests, and it's going to be falsifiable, which means we have to be able to say that this hypothesis is incorrect or this hypothesis is correct. If your hypothesis is incorrect, that does not mean you failed at your experiment. That means that what you predicted didn't happen. And that's okay. That means you learned something in the end and you can tell me why it didn't happen. This didn't happen because blah, 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 blah. And I want to know about the blah, 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 blah. Okay? So you can't prove conclusively or as a whole that a hypothesis is true, but the more evidence you're going to gather in support of it, the more likely it is to be true. If you cannot test your hypothesis by making an observation, it is not scientific. These observations can be direct, like something you see with your eyeballs, or indirect, something you use like a microscope for. And what is an observation? An observation is any information you can gather with your senses. That means vision, hearing, touching, smelling, tasting. Okay? Besides raising questions for investigation, observations play another role in scientific investigations. They're going to help us to gather evidence. So we're going to write a hypothesis based on what we did earlier. So how the amount of sunlight is going to affect the number of flowers grown and why we think this is going to happen. So we're going to write hypothesis in the format of if independent variable happens, then dependent variable occurs because of some prior knowledge that we have. So I can say, I'm going to write this up here at the top. Oops, that's not what I meant to do. If independent variable, so if a rose bush receives, ooh, my handwriting is bad. Come on. Wow. More sunlight. Oh my goodness, I do not know what's going on. Hold on just a second. So if rose bush gets more sunlight, let's try this again, one more time. If a rose bush gets more sunlight, comma, then it will grow more flowers because plants require sunlight for photosynthesis. Photosynthesis is the process by which plants grow, by the way, if you didn't know that. So is this testable? Can I put a plant in sunlight and see how many flowers it grows? 100%. This is testable. This is something I can see happen. Is this falsifiable? Of course. If my plant in the closet in the dark grows the most flowers and doesn't receive any sun, then obviously this is not true and it's falsifiable. So that is how that works there. So let's see what hypothesis I wrote when I did this earlier. I said, if a rose bush receives more sunlight daily, then the rose bush will grow more flowers because sunlight is vital for the process of photosynthesis. So there you go. You guys, if you have any questions, please feel free to message me. You're going to do great.